Okay, I'm going to get started. So it's my pleasure to welcome you to the inaugural QBI Weizmann Institute Symposium on Molecular Systems Biology, which is being co-organized by myself, Tanja, Saki, Noam, and Dani. And it's a multi-part series. This is the first part. We're going to have the second part a week from today, the same time, same place. And one of the goals here, I would argue, of this particular symposium is to enhance collaboration between these two institutions. And obviously there's a lot of collaboration ongoing already. It's to further enhance that collaboration, and it's also to initiate new collaborations. And I would argue that the scientific vision between the two institutions is incredibly synergistic. And I'd also like to say that it's a set of people that are very fun. So I think that's another big plus of putting together these two groups of people. And maybe I'll let Saki just say a few words about this relationship and what it means to the Weizmann Institution.
Thank you, Nevan. It's really a pleasure to open this meeting, done in an unusual setting, unfortunately. I still remember that you invited us to come over to San Francisco. This was going to be a physical meeting before COVID changed our plans. But nevertheless, here we are. And indeed, we celebrate the commonalities and the mutual interest and friendship and collaborations that we already have. And we're looking forward to building them and expanding them much further. So the community of systems biologists at Weizmann is very active, fastly growing. The QBI is similarly active and leading in systems biology. And it's only natural that we will collaborate together. It reminds me that about a decade ago, we did something similar with Harvard Systems Biology at the medical school. Actually, almost the whole department came over to Weizmann from Boston. And that definitely led to many mutual interactions between the two universities, between the two institutions. So I look forward to those 2 days and to what will follow, which will definitely be exciting and unpredictable even. So let's just move to the science. It's going to be exciting.
Thank you, Saki. Hopefully unpredictable, for sure. So over the next couple of weeks, we have 8 speakers from both institutions that'll be presenting some of their work. So today we'll have 4 speakers from QBI UCSF and 4 speakers from the Weizmann Institute listed here. And then, as I said, a week from today, we'll have the additional 8 speakers, again, 4 from each place. And the third session, which will be 2 weeks from today, will be a discussion with the PIs on how best to go forward. And I just have listed here the speakers, but this is obviously open to any PIs at the Weizmann Institute or those affiliated with QBI at UCSF. And the discussion will be about how to go forward scientifically, but there'll also be a discussion on how to potentially philanthropically raise money to support the collaborations that come out of this a relationship. And on that note, I just wanted to get Andy Weissman here to say a few words, who is hopefully going to help us out on that front. And Andy is the executive director of the Bay Area program of the American Committee for the Weizmann Institute of Science. His role, avocation, and dedication is to raise funds for an awareness of the Weizmann Institute and its core mission of science for the benefit of humanity among major philanthropists and philanthropic organizations. So Andy, if you could say a few words about this particular relationship, that would be great.
Sure. Thanks, Nevan. Thanks everybody for being here today. When I first heard a while back that this has been kind of percolating for quite a while amongst the scientists, of course the development guy would say, well, let's see if we can get it moving forward. So I reached out to Nevan and Jacqueline back in February, and then a small thing happened to interrupt the world, and COVID-19. And so we put it on the back burner, but then once Zoom became the mode of communications and interactions, we started talking again, and boom, all of a sudden Jacqueline and Nevan were on the phone with Danny Saki, and our VP at Weizmann. So off we go. So the important thing to know about this is that for Weizmann, in terms of philanthropy in the Bay Area, we do fairly well here. But we also are an organization that's 7,500 miles from any of the peers or any of the really good hotspots for food and science. So what we're trying to do is see if there are synergies and natural connections amongst scientists, amongst people who are from QBI and Weizmann, to see if there are collaborations that indeed can be robust and questions that you can each ask each other and rely upon each other for support. And so my job is not to be involved in this part at all. I'm just going to be a listener, of kind of a fly on the wall. But once you go through this exercise and you have these discussions and things bubble up to the surface where you think that there are collaborations that can be robust and helpful to what you want to do to advance science, that's when I will step back in and talk amongst the philanthropists and organizations here in the Bay Area to help raise funds for you, so you don't have to only go out and look for these scientific grants. So that's what I'm here to help. But the important thing for you to know is that as development people and fundraisers, we don't want to be the leaders. We want this to be from the bottom up, from the labs and from the groups in both organizations to come together and in a very natural way. And so I just wanted you to know that we're here to support you. I look forward to working with you all if something comes out of it that you think you need my help on. And I'm very excited that we got this started. So good luck with today's symposium and those coming in the future.
Thanks, Andy. That's great to have you on board. Andy told me previously that it would be very hard not to raise money for this group of scientists in the Bay Area and in the Weizmann. So we look forward to working with you in the future. And just a few words about how this started. Saki had mentioned a little bit about this. So there was a number of us that were actually in Tel Aviv for another event, and there's a group of us here from QBI UCSF going out for dinner. And I think every one of these people at this dinner table actually also gave a talk at the Weizmann Institute, because if you come to Israel, you have to go to the Weizmann Institute. And actually, I ran into Jamie there as well with Danny, and this is a phrase that James gave to me. I'm going to let Saki, can you interpret that for me? I want to make sure what Jamie told me was accurate.
I would need help from somebody.
Lumberjacks, Saki.
Exactly. Can you help me here?
2 Canadian lumberjacks.
Okay, that's better than I. That's 2 Canadian lumberjacks and Danny. So that's better than what Jamie told me this was going to say. So that's good. So, so there's a lot of relationships already, obviously, between QBI, UCSF, and the Weizmann Institute. So we're just leveraging what's already in place and just building upon it. And QBI has also been at the forefront of international collaborations around the world, in Europe, in Africa, in Israel. And one of the things that we're involved in to facilitate collaborations is this symposia series. And this was what QBI had planned for 2020. We had approximately one a month that we were going to do, and we actually got through the first 2, and then the pandemic hit, and then this forced us to pivot and to go virtual and go online. And this is exactly what we're doing here with the Weizmann Institute. We had a couple of weeks ago a very successful symposium on COVID-19 as well. So we're pivoting, and we're going to have just as a robust symposia series going forward, at least for the next year or so, online. So please look for these. And the people that deserve the most credit for this particular event and all the events that we do are these individuals. So the Chief Operating Officer of QBI, Jacqueline Fabius, and our great media team, Gina, Alexa, and Carolina. So thank you guys very much. And just one last word here about the Quantitative Biosciences Institute. It's very much about collaboration. It's about bringing people together from different disciplines bringing together technology, both experimental and computational, together in a disease agnostic way and focusing it on a variety of different disease areas. We're into investing in the young. We have a number of RFAs that we put out in collaboration with other entities on campus and elsewhere. We have a strong media outreach, seminar series. I talked about the symposia. And we've also started these different cell mapping initiatives. And this is going to be a segue now into my presentation, where we've taken these disease agnostic tools and tried to put them together in a pipeline and apply them to different diseases, including cancer, infectious disease, and psychiatric disorders. And just before I start my presentation here, I just want to remind all the speakers at the 8-minute mark, 10-minute talks, at the 8-minute mark, there'll be a sound. Carolina, do you want to just quickly give that sound? Well, there'll be a sound at 8 minutes, and that'll give you a signal that there's 2 additional minutes, then we'll have 5 minutes for questions. Okay, so here is a big part of the scientific vision, I would argue, of what we try to do is we try to take different technologies and put them together in an integrative way, in a disease agnostic way, and focus them on a variety of different disease areas to try to get at the underlying biology behind these different disease areas. So we're heavily involved in putting together mass spectrometry-based approaches with CRISPR-based genetics, structural biology, which you'll hear a lot about today, as well as chemical biology approaches. And we put all these together and focus them on these different disease areas in the context of the Cancer Cell Map Initiative, the Psychiatric Cell Map Initiative, and then the Host Pathogen Mapping Initiative. So we've been focusing on a variety of different pathogens over the last several years. And I would argue we've been in a perfect position to pivot and expand now to focus on COVID-19. And this is what I'm going to give you a brief update on as the efforts of the QCRG, the QBI Coronavirus Research Group, which is recently formed. It involves over 40 different laboratories affiliated with QBI and UCSF, involving essentially hundreds of different scientists. And we've actually broken up this larger effort into 12 different subgroups focused on different technological focuses, such as structural biology and drug discovery, and then also focuses on different biological areas that the virus comes in and hijacks during the course of infection. So we put these groups together that are interacting very successfully, again, involving hundreds of different scientists working together, I would argue, in a very exciting and unprecedented way to try to come up with solutions for this COVID-19 pandemic. And I just want to briefly tell you about 2 papers that have recently come out from the QCRG. One focused on protein-protein interaction mapping, looking at the SARS-CoV-2 proteins, and the other globally looking at phosphorylation, changes in the host in the context of infection, but both focused on trying to understand how the virus comes in and hijacks and rewires the host during the course of infection in an effort to come up with host-directed therapeutic approaches to help fight SARS-CoV-2. So first here with this paper, so we generated a protein-protein interaction map using a systematic affinity tag purification mass spec-based approach. Here's a first look at the protein-protein interaction map, which many of you obviously have been exposed to and are digging into, where the red diamonds here are the SARS-CoV-2 proteins, and then the other ones, the other nodes are human proteins. If the node is orange, we argue that it's a potential drug target. And I just wanted to point out that we've collaborated with Zoic Labs. It's a Hollywood company that's actually involved in making movies. They've been involved in making Life of Pi and Titanic. And Biz Stone, the founder of Twitter, and Ron Conaway got us connected with this particular company. And they've done some very interesting things in terms of visualizing our data in a very interactive and intuitive website and database. So you're interested in our data, I would encourage you to look at this particular website, and we're continually putting more information in here. So one of our goals here was to try to find human proteins that would be targeted by a drug or a compound, obviously for another disease that could be used in a drug repurposing effort. And this is work that was spearheaded by Brian Shoykat and Kevin Shokat. They looked at our maps and they came up with 69 different candidates, a mixture of FDA-approved drugs, drugs in clinical trials, and preclinical compounds that would target at least one of the host proteins that we think the virus would need to infect our cells. So this is the one drug that's obviously being used, remdesivir, targeting NSP12. Then the question is, are any of these other drugs and compounds, could they have potential antiviral effects? And we've been collaborating with groups around the world, including scientists at the Pasteur Institute in Paris and in New York at Mount Sinai Hospital, to test these. And we've narrowed in on about 10 agents, which we're very excited about, which I just want to briefly tell you about. A couple connected to protein translation, including zotadafin. This is a protein that targets EIF4A, a translation initiation protein. We found that its interacting partner, EI4H, interacts with NSP9, a cornavirus proteins. That was our connection here. And this is from the company Effector that was started by Nevan Krogan and Davide Ruggiero. This has potent antiviral effects. We're working with Effector to get this into a clinical trial. There's another inhibitor of protein translation elongation. This is a compound synthesized by Jack Taunton, ternatinin-4, which has antiviral effects. It targets, interestingly, a protein involved in translational elongation, which is also targeted by a structurally unrelated protein called pladepsin or aplidin. And this is from a company in Spain, which has actually now taken this compound or this drug into clinical trial for COVID-19, and we're interfacing with them right now. So that's one group that we're excited about. This is being led by Nevan Krogan, and then the other group, which is being led by Brian Shoykat, are modulator of sigma R1 and sigma R2. These are two receptors that are still classified as orphan because you don't know what the natural molecule is that comes in and binds it and manipulates it in our cells. But we found a number of sigma modulators that have antiviral effects, including antihistamines, the female hormone progesterone, and a number of preclinical compounds as well. And if you just looked at the chemical structures of these, there's no way you could group this together. But if you know what the target is, which we think we do, that's how you group these together. So this is a great example of knowing the biology. It's so much more powerful when you go to the chemistry and the pharmacology. And Brian's now augmenting our search space and trying to find kind of a best-in-class molecule or drug in this to take ultimately to the next level. So these are two groups that we're very excited about from this initial study. And then just very briefly to tell you about the next study where we've globally looked in human cells and Vero6 cells, looking at phosphorylation changes in the context of infection. And this is important for a couple reasons. We're trying to identify the kinases that are misregulated. And if you think about it, the kinases are like the master switchboard of the cell. If you could identify which ones of these the virus needs to manipulate our cells, this would be very advantageous. That's one important point. And the second important point, as we all know, kinases are some of the most druggable proteins in our cell. So there's a lot of pharmacological inhibition that can be carried out to see if they have any antiviral effects. So we globally looked in host cells at changes of phosphorylation. This was work done in collaboration with Paris. So 2, 4, 8, 12, and 24 hours post-infection, we're looking at phosphorylation changes. And then working closely with Pedro Beltrao in Cambridge at EMBL EBI, he looked at the phosphorylation changes and then identified, working backwards, which kinases were either upregulated in red here or downregulated in blue. So in total, we actually identified about 47, approximately 50 kinases that we think the virus is manipulating. And if you look at the entire kinase repertoire, there's over 500. So about 10% of the kinases we think the virus is hijacking during the course of infection. Well, then we can work with people like Nevan Krogan at UCSF and then identify the kinase inhibitors that are manipulating the kinases that the virus is manipulating. And then we could check to see if they have any antiviral effects. And we found a number which we're very excited about, including casein kinase 2 inhibitors. Casein kinase 2 is actually the most upregulated kinase. If you look over here on the heat map, CDK inhibitors, PI5 kinase inhibitors like aplamode, which we're very excited about as well. And then we actually found a number of kinases in the P38 kinase cascade right here in the middle, all these MAP Ks that all work together in this P38 pathway. And we found a number of inhibitors of this particular cascade that actually have antiviral effects as well. And then also working with Marco and Veronica at Paris, we could manipulate genetically using RNAi these different MAP kinases and find they also have antiviral effects, confirming that our pharmacological inhibition of this pathway has antiviral effects that we're looking into in more detail. So this is a set of drugs that we're looking into. And there was a recent piece in the Chronicle which outlined some of our work and some of the drugs and potential clinical trials that are starting from our work. And there's a list of 10 here that we highlighted. There's a number that are actually being started clinical trials based on this work. And I found out last week, there's actually 3 more clinical trials that are starting based on these kinase inhibitors. And we're working closely with the companies that are carrying out these trials. So we're very excited about this set of drugs and trying to take them to the next step. So future work, as I said, transitioning this work into clinical trials with collaborations with people around the world. I'm very excited about this combinatorial work. I really think that one's going to need a cocktail approach to come up with to combat COVID-19, similar to what's being used for HIV. So we're doing a lot of combinatorial work in the laboratory, combining many of our drugs with remdesivir and combining them with each other. Also systematic genetic studies using CRISPR and RNAi to confirm the genes that we're identifying proteomically do have antiviral effects. And then we've just actually finished now a protein-protein interaction map of SARS-CoV-1 and MERS, and we're now doing evolutionary comparative studies of SARS-CoV-2, SARS-CoV-1, and MERS to get at more of the underlying biology behind why these different viruses cause trouble and why they cause trouble in different ways. So this is a work to come very soon. So this is a picture of my group that was taken, I think, a couple months before the pandemic. Just to highlight a few people, there's David Gordon and Gwendolyn Zhang. They led the charge in terms of the protein-protein interaction work that I briefly alluded to. Here's Mehdi. He was the first author on the phosphorylation study that was carried out. And just to highlight a few other individuals that lead the mass spec facility affiliated with the group, Danielle Swanee, Ruth Huttenhain, and Robin Cake. These are fantastic scientists to be working with. These ones I pointed out, and the whole group, and all our collaborators as well. So this is just a brief summary of some of our efforts here with respect to COVID-19. And I thank you for listening. I'd be happy to take any questions right now. And I'll remind all the participants to please put their questions in the chat box so that they can be asked of the speakers. Okay, I have a question. I'll just ask it myself, I guess, here. How similar are the protein-protein interactions of CoV-1 and CoV-2? That's from Tanya. They're actually quite similar. The genomic architecture isn't identical between them. There's some proteins that exist in SARS-CoV-1 that don't exist in SARS-CoV-2 and vice versa. But overall, they're actually quite similar. Obviously, the differences would be very exciting, and we're digging into those right now. But where you see a much bigger difference is between MERS and SARS-1, SARS-CoV-2. So we're implementing in this differential interaction scoring system that we'd actually developed with Trey Ideker to look at cancer here. And this data will be, we've finalized this data as of, I think, yesterday. And it's very exciting, and we're figuring out how best to visualize it as you compare across these viruses. So Tanya, if you want to see this data, we'd be happy to send it to you, or anybody else on this call. We'd be happy to send you this information. Okay, so maybe we'll, if there's no more questions. Oh, Saki, you have one?
Shall I read it aloud or?
Sure.
So I'm wondering, the targets that you're seeing, in which tissues and organs in the human body are they expressed, and do they tend to be relevant to body sites where COVID is infecting?
Yeah, that's a great question. So the initial maps that we generated were created in HEC293 cells, which are obviously kidney cells, and it was not thought that COVID-2 is affecting kidney, although it seems like it's affecting every organ now. But when we looked at our set of initial 332 human proteins derived from kidney cells, what Pedro Beltrão did was just globally look at all tissues in terms of where these 332 are most highly expressed, and across essentially all organs. And what he found was that despite us collecting this data in kidney cells, where these 332 human proteins were most highly expressed were in lung cells, which I thought was very interesting, which gave us more confidence in the data that we're ultimately generating. That being said, maps are now being generated, and data is being collected, obviously, in a number of different cell types, including A549 cells, lung cells, and cardiomyocytes, and other cells as well. So it'll be very interesting, not just to compare across different viruses in the coronavirus family, but different cell types in the human body, as well as different species. Actually, Adolfo Garcia Sastre just sent us some bat cells, and we're going to be collecting some of these protein-protein interaction data sets in bat cells to try to give us some insight on the process of zoonosis. Is there anything else, Saki, or do you think we should move on here?
We could, or maybe wait, maybe invite more questions. I don't know if there are any. Anything that targets ACE2 by chance?
Yeah, I mean, the way, we've looked at spike interactions, and we have a couple of interesting ones. But what I'll say is the way we've set things up, we're really looking intracellular for those interactions. We are now going back and looking at the cell surface. We're setting up a protocol to look for interactions at the cell surface, and I think this is where you get some interesting interactions with spike and ACE2, and who knows what else.
Yeah.
Maybe I can add to this targeting ACE2. There's a number of really interesting effort in QCRG at UCSF using proteins to target ACE2. And so we have a number of different variants, both from a selection as well as from computational protein design from my group that actually neutralize in assays.
Thank you, Tanya, for bringing that up. So there's a big push, not just for drugs, but also biologics. And obviously a big focus is on the ACE2 spike connection in that regard.
Yeah, makes sense.
Okay, so maybe we'll move on here to the next speaker. That's Roy Abraham from the Weizmann. He's in the Department of Biological Regulation at the Weizmann Institute of Science. And he's going to be talking about predicting human infection outcome using dynamic deconvolution of single-cell RNA-seq data.
Hi, good evening, good morning. Thanks a lot for the invitation. I will just go right into the presentation. So we are, in the lab, actually, we're interested in bacterial pathogens. This is not hype right now, I know, but I would claim that the fundamentals are the same. Specifically on intracellular bacterial pathogens. And one of the main things that is obvious during infectious disease is that if you're exposed to a pathogen, not necessarily you will get the same outcome from the infection. Either you can become disease-free or have some active disease that can have mortality implications. And really the question I think in the field currently is what are the differences between these individuals? And if you stratify infection as on the x-axis as time over bacterial load, then you can follow these different outcomes over time, where you can see that you will have an initial increase in bacterial load in most people, but then they will clear and become disease-free. Some people will develop latency or latent infection, which I will discuss later. And the people with active disease, and our claim in the lab is that most of the studies will focus at late stages of infection, where already the bacterial load is very high and anything that the immune system has done has already failed. What we are trying to do in the lab is to focus actually in these early stages where we think there is a decision point that is actually crucial for the outcome of the disease. In the lab, we use blood immune cells in order to look at these early stages. This is available always as a window to the immune system. And we use single-cell RNA-seq of the blood immune cells during the early infection stages in order to see whether we can find predictors of the outcome of infection using these signatures. And I will show you some examples for this. So these are the people leading this project, Shelley, Noah, and Leah. And mainly what we are trying to do is to use ex vivo infection of blood immune cells to find out what is the repertoire of immune cells and their specific responses. How do they respond to different pathogens? And these are usually the model system we use, Salmonella, Listeria, and Mycobacteria. And to understand at the end, what are the universal responses to infection, but what are also the individual variation between individuals that we claim would define the outcome of infection at later stages. So I will start with giving you an example of how does an ex vivo infection look like. So we infected blood immune cells of healthy individuals with either Salmonella or Listeria, and this is how the data looks like. So these are the single cells. Cells either before or after infection, they like to stay together if they are from the same cell type. So you can actually annotate the cell types of these different cells, as you can see here. And you can start to tease out what are the differences of the responses between Salmonella or Listeria of different individuals. As an example, I give you here NKT cells, which respond mainly to Salmonella infection. And what is interesting about this is that interferon gamma, one of the main cytokines important for intracellular infection, is secreted only during Salmonella infection. We can go even deeper and look at the different Salmonella strains that you get, either Typhimurium, paratyphi, or typhi. And you can also profile the responses of the blood cells to those. And what you find is that actually there is a specific Salmonella response to a specific Salmonella strain that is elicited by these NKT cells. You can further characterize this way the pathogen-specific and the cell-to-cell communication that are specific to each pathogen, or the universal responses. But as I alluded before, what we actually want to do is to look what happens in patients as a step towards that. What we wanted to look is to understand is how does in vivo infection looks like. This is not an easy task, and as you might expect, how do you look at early events of in vivo infection? And for these, we teamed with a group in Oxford that they actually developed the human challenge model. They give individuals Salmonella typhi, and they follow them for 7 days. And we collected blood from these individuals, these volunteers, at different time points after infection. And we profiled the blood, in vivo blood responses to these to this infection. We can also stratify what is the outcome of each of these individuals at the end. So some will develop typhoid disease and receive antibiotics, and some will remain asymptomatic, so no typhoid disease, as denoted here by anti-BNT. And when we profile those 6 individuals at 3 time points, we can stratify the single cells based on their time point after infection. So T0, 12 hours after infection, or 7 days. We can stratify them based on whether they developed disease or did not develop disease. As you can see here, there's several responses or several cells that are correlative to each of these phenotypes. And we can also look at the cell types that are lying behind these different responses. We can further classify them to subtypes of monocytes, T cells, B cells, and so forth. But I think that what is interesting is if we look at the dynamics of these responses, we can actually see that even with these 3 versus 3 individuals, we can see a response that is unique to the typhoid disease versus the non-typhoid disease. Of course, this is not great for statistics, 3 individuals versus 3 individuals, and we really wanted to take this a step further and to understand how can we do this on hundreds of individuals. And there's a limit to how much single-cell RNA-seq you can do on hundreds of individuals. And at this point, what we developed is an algorithm that allows us to actually extract the cell type and the cell type specific immune response from the single cell data that we already collected and infer similar information from bulk RNA-seq. This is an algorithm that was developed in the lab. You are welcome to take a look at it. It was already published. What we basically do is we take the single cell RNA-seq, we convert it into 2 vectors. One is the vector of genes that correspond to the cell types, and one is the vector of a pathogen-specific and immune cell type-specific infection-induced state. And we can extract from these 2 vectors basically the bulk data that they represent. So what we can do now is take bulk RNA-seq data from hundreds of individuals and infer the same kind of information exactly. And as one example of this, I'm going to delve into TB disease. If you're not familiar with Mycobacterium tuberculosis, so this is a disease that is considered to be inside a third of the human population. So a third of the human population is considered to be infected with TB. But only 5 to 10% will actually develop this disease. Now, what we will be interested in is to understand what would be the impact of the initial contact of the patient with the first exposure to the pathogen until the appearance of symptoms. And here also what we will do is apply our single-cell RNA-seq method. But in this case, I will show you how the deconvolution algorithm really performs when applying it to bulk RNA-seq data. Here again, in order to really understand what goes on at early stages of infection, what we did is we collected, of course, with collaborators, we collected a cohort of individuals that were exposed to TB. So once you're exposed to TB, this is a highly contagious pathogen. Once you're exposed, you are considered infected. Then they followed up on these individuals for 2 years. And over time, you can see that some of these exposed individuals actually developed disease. So all the individuals are considered latently infected, and some of the individuals that develop active disease and symptoms will be considered progressors. And we applied bulk RNA-seq data on this longitudinal follow-up study and applied our deconvolution algorithm. I will show you just a hint of a result of one of the signatures of one of the cell types during the course of this disease. So on the y-axis, you will see this index of monocyte activation, a specific signature of monocytes during using the deconvolution. This is how the controls individuals look like throughout the longitudinal study. So these are months, okay? These are months. You can basically average them and see how the controls like over time. And if you follow one of these progressors, so latent to active, you can see how this signature starts like, and you can see that this signature actually increases during time. What is interesting, and if you notice here, is that this signature actually falls when you get to the active disease. And this pattern was obvious in all the patients that we collected data from that progressed from active disease to, from latent disease to active disease. As you can see, all of them displayed heightened activation of this signature even before any symptoms appear. And the implications for this for treatment at the end of the day is huge, because if you think about it, if you can start treatment already before symptoms appear, that can have huge impact on the progression of disease. So with that, I will just summarize that what we are trying to do currently in the lab is expand our knowledge base or expand our database of different responses by different individuals to pathogens, either ex vivo in healthy cohorts or in vivo infections. From this, we extract the signatures using our deconvolution algorithms that allows us to look at cell type abundance, cell type activation states, and pathogen-specific responses. You can imagine that we can now look at different responses to different pathogens to the blood and extract this information from bulk RNA-seq data. And hopefully, this will allow us to extract early biomarkers of infection. At the end, what I believe is that we can use that information to offer better treatment options. So with that, I want to conclude. I want to thank all the lab members, of course, and our collaborators with whom this work will not be possible, and the funding. And thanks again for the invitation.
Right, very good. Thanks, Roy. I got a couple questions here. One that was just texted to me. Are there environmental factors that you think are going unmeasured that would influence these signatures?
Definitely. So one of the interesting things is of course, the differences between the individuals, the variation between individuals, and definitely there's environmental factors that going into there. At the end of the day, what we want to be able to is to look above those environmental factors and really understand what's going on between the different individuals. Definitely this will have a signature on the immune response.
Another question, have you looked at it's related, I guess, have you looked at the formation of an adaptive response, such as an exhaustion marker?
So, you know, this is an interesting point. At the end of the day, I think that the adaptive response definitely is important for the ability to control the pathogen. You know, unlike viruses, there's a huge difference in the adaptive response between viruses and bacterial pathogens. for TB, for example, the adaptive response is considered to be very different than what you will have. There's no B-cell immunity almost. So I would claim that, yes, this is a factor. This is a factor also that we are trying to think as preemptive as we can to look at, and not at the point where the adaptive immunity kicks in.
Maybe one more question from Tanya. She's asking about some biology. What's the most exciting biological hints that are in the signatures for you?
I think that the most exciting ones, and I think the ones that are most surprising to me, is the fact that we can actually tease out differences in the immune responses to different pathogens. I did not expect that. So, for example, there's been a lot of hype about the type 1 interferon and COVID, and it's type 1 interferon has been published already in the context of many infections. We see exactly the same response in Salmonella typhi infection. And I wouldn't like to use that as something that I would call a signature for COVID. But we see actually really specific responses to different pathogens. So for example, the NKT response to Salmonella, and I can already say that we can see Listeria-specific responses, TB-specific responses. So to me, this was a real surprise how vivid those differences are.
Right, awesome. That's great. Great stuff, Roy. Okay, we're going to transition to the next speaker from the Bay Area, Andre Shally, coming from his house in Burlingame. Andre is the Associate Dean and he's Professor in the Department of Bioengineering and Therapeutic Sciences, and his talk title is Integrative Structural Biology.
Hi, so thank you for having me, first of all. What I will describe is a short outline of a collaboration we have primarily with Jan Ellenberg's group at EMBL. But as always, on anything to do with the nuclear pore complex, which this project involves, we also are in touch with Mike Kraut and Brian Chait at Rockefeller University. And the main person doing the work in our lab that I'll talk about is Jeremy Temkin, a great postdoc in the group. So in our group, we are concerned with integrative or integrative structural biology, which is illustrated here on this slide. Integrative structure determination is the type of structural determination that relies on, in principle, all types of available information about the system that we wish to model structurally, regardless of whether it's coming from this or that experiment, physical theory, a statistical inference, or some prior models. It's in principle a good idea because by maximizing information used, one maximizes the accuracy, precision, and completeness of structure determination. And the way it's formulated is as a search for all models for which computed data match the experimental data within some acceptable threshold. It's been reviewed and applied many times in the past. It's illustrated by its recent application to the nuclear pore complex, shown here at the bottom, based on the various types of data that you see listed on this slide. I shall not go through this list. What I'd like to emphasize here is that so far, this approach has been applied to determining static structures, and at most, mixtures of static structures, of a few static structures. But all biology is dynamic, or almost all biology is dynamic, and what we'd like to do is generalize this approach so that we can determine models of dynamic processes based on all information, whether it's from experiments or physical principles. And I will illustrate how we are going about that today with a short vignette. With an application, the development and application of a preliminary version of spatio-temporal integrative modeling to the modeling of the assembly, the process of assembly of 550 or so proteins of 30 different types in the nuclear pore complex during the cell cycle in the context of the nuclear envelope. The scheme of this process is indicated here for the human cell in particular. There's different two different stages at which the nuclear pore complex assembles and falls apart. It's a very important process in the cell cycle, so it's important to study it biologically. Now, what we will do is use different types of input information, as I said, as always in modeling, in order to construct a model that's consistent by that input information by construction. And I'll highlight on the next two slides two main types of input information that we rely on. First of all, we have the structure of the endpoint. We have the mature yeast structure that I already mentioned, determined through the static integrative structure determination for yeast. It's kind of decomposed on this slide. There's different modules in this structure highlighted on this slide that together form the entire nuclear pore complex in yeast. Now, there's a little bit of a problem. Jan is studying the human structure, the assembly of the human nuclear pore complex. We have the complete structure of the yeast NPC, but no worries, there's a partial structure of the human NPC generated by Martin Beck, also at EMBL, and by putting the two together, we can get a model of the human NPC that's essentially complete. So we do have a nearly complete model of the human NPC structure as the endpoint of the assembly process, as the first major input data for our modeling of the process of assembly. We have, very importantly, two more key data sets. The second one is a spatiotemporal kind of a data set about the progress of assembly of the human NPC, namely through lifestyle correlated electron tomography experiments, which Jan's group has already published. We have information about how the shape of the complex, including the nuclear envelope, as well as the assembling proteins, changes as a function of time during the assembly process, which lasts on the order of tens of minutes, so it's possible to do such characterization. We also have, so far, unpublished data about complementary data about how the composition, the copy numbers of the, in fact, the stoichiometry, I should say, not just the composition of the constituent NUPs, the proteins that comprise the NPC, how that changes as a function on time on the corresponding time scale to the characterization of the shape by electron tomography. So these are going to be two major data sets that we wish to take into account while we are constructing the model of assembly, not just to check the model of assembly, to increase the odds of coming up with a model that's more accurate, precise, and complete than it would be otherwise possible. And so here's the key idea. It's actually very simple. What we'll do, we'll model several, let's say on the order of 8 or so different snapshots during the assembly. And then we will connect using the information that I mentioned. And then we will connect these snapshots in a sensible way to create trajectory models. And that'll be then a set of such trajectories that are sufficiently consistent with input information will be our model or ensemble of models, if you will, of the assembly pathways. A little more precisely, I cannot dwell on the details. The model of the pathway is in fact a probabilistic one. We take a Bayesian view. We define the joint probability of the structure and composition at any given time snapshot on the trajectory as the posterior model density that we wish to define based on input data and then sample in some fashion in order to determine the final model. This joint probability density function is obtained by multiplying independent data likelihoods, each one of which depends on various types of input information, most of which I already outlined, and that's enumerated on this slide. So we rely on the knowledge of the final structure, we rely on the shape from tomography, we rely on the composition, stoichiometry from microscopy. We also have the shape of the nuclear envelope, not just the NPC as a function of time. And this all contributes to the modeling of the individual snapshots using standard integrative structure modeling that we've applied many times before. But now we just do it for each snapshot. We do this and we get a bunch of good scoring snapshot models, partial structures, evolving towards the mature pore at each of the discrete times along the assembly, and that's schematically illustrated here. Now, we could, in a simple prediction, just take the most likely, the most satisfying model from each of the time point and connect them together to get a reasonable prediction of the assembly pathway. But that's not all that we can do. We can also imagine having information about transition probabilities between different states. There's multiple possible states given our information at each stage. And so there's different interconversion probabilities we can get from different sources. And we
Andre,
those.
2 minutes.
Yep.
2 minutes, Andre.
Yep, thank you. And we can consider these transition probabilities in addition to snapshot probabilities to obtain a more complete and more discriminative path, entire total pathway probability. Some of the possible sources for these transition probabilities include various theoretical considerations, such as pairwise structure similarity. Two similar structures are more likely to interconvert into each other than two different structures, or more physical simulations, such as Brownian dynamics simulations could also generate such transition probabilities, or they could come from data measurements if we had such measurements. So far, we applied structure similarity-based transition probabilities only. We sampled the entire pathway posterior model density with a Monte Carlo process, and we obtained an ensemble of good scoring pathways. This is the actual notation of that. Looking like the one here, the assembly pathway progresses from left to right to a single structure on the right between specific states in a specific order. And we could then look at this ensemble in various ways. And in general, what this model and ensemble of pathways reveals is probabilities of different molecular events, which you get out of their likelihood in the path ensemble. And what this shows here, just to illustrate without going into details, is the best scoring pathway and the sorts of things that you can learn about the order of which proteins comes before other proteins and so on. And that's zoomed into on the next slide that shows a prediction from the model about the order of one particular protein joining the complex, preceding two other proteins. And that's the sort of a prediction that can be very directly validated by the experiments that Jan Ellenberg can actually perform in his lab, as illustrated on the slide, and that's work in progress. So in summary, I illustrated the idea and preliminary implementation of Bayesian integrated modeling of dynamic processes as opposed to static structures that's based on data as well as prior models of dynamics. It's important because biology is mostly a movie, not photographs. The method produced the model of the assembly pathway of the NPC in a preliminary fashion, modified the model, satisfied the input data, and there were testable predictions based on that model, as every model should be asked to do. And that's it. That's my 11 minutes. And I end acknowledging the collaborators again as we started. Thank you.
Thank you, Andre. Fantastic. So a couple of questions. One from Danny. How are these complex, multi-component complexes assembled in a hierarchical and time-dependent manner? Can just different K-on, K-off rates account for, or are there other tricks? Danny wants to know about the tricks.
Yeah, that's obviously a great question. You know, we are playing with the idea of exploring how far we can get by based on the knowledge of currently synthetic or invented association and dissociation rate constants between different pairs of proteins in the system. But I have really nothing particularly insightful to report yet on this front, other than it's a great question. We're looking in this direction, and that's about all I can say. There's actually, I'll say one other thing. There's actually interesting hierarchy here, right? Because proteins themselves assemble through interactions between atoms, just like protein complexes assemble through interactions between proteins. Perhaps there'll be a lot of inspiration from what people have done for protein folding also here in the area of protein complex assembly.
Another question, at what point does computational cost become limiting? How do you balance detail versus completeness?
Yeah, of course. So, you know, what that's also a big question that's an ever-present question in modeling in general. In general, what we invent the representation of the model out of thin air based on our experience. That determines the detail at which the scoring function is defined, as well as the detail at which the sampling needs to be performed. And what in general we try to do is come up with hope that there is a representation that is sufficiently detailed to result in a model that is useful for answering the questions we are interested in, while at the same time also has a sufficiently accurate detailed scoring function, while also at the same time is sampleable, is sufficiently simple that it can be sampled well enough with the existing computers and sampling algorithms, or at least algorithms we can come up with. So it's there's no guarantee there is such a representation exists, but that's what we're trying to do each time. We have also formally explored this issue a little bit more deeply by asking what's an formally optimal representation for integrative structure modeling in a paper in PNAS last year, I think it was, where we defined an explicit criterion of optimality of representation based on the considerations I mentioned. And there we explicitly optimized for representation that was then used for modeling as you know, an exploration of this a very good question again.
So there's more questions in the chat, Andre. I'm going to ask you to answer them. One from Denitsa, one from Onita. So, but they're great questions, but we're going to move on to the next speaker. Noam Stern-Ginossar from the Weizmann in the Department of Molecular Genetics, and she's been doing some really exciting work, I would say, tracking COVID-19 across Israel. And her title is The Coding Capacity of SARS-CoV-2.
Thank you, Nevan. So I'll start by introducing my lab with a few words. So my lab is interested in viruses and how they interact with subvert gene expression pathway in infected cells. And, oh my God. Okay. And we mostly study herpes viruses, which are large double-stranded DNA viruses that replicate in the nucleus. They infect probably all vertebrates and even some invertebrates. And one of the really unique and defining features is that they persist for the lifetime of the host they infect. So you never clear a herpes virus infection. And this shows us how well adapted these viruses are for their host. And this adaptation was crafted along many evolutionary years, making them a really strong tool to study viral-host interactions. So generally in my lab, we follow 3 different schemes. On one, we look at the changes in post-transcriptional gene regulation during herpes viruses infection. We are also really interested in understanding herpes viruses latency and actually what governed the molecularly, what governed the infection outcome. Why is one cell infected latently and the other infected latently? And the third route, which is related to what I'm going to talk about now, is to uncover the complexity of herpes viruses. And now we use our expertise to actually extend it to the new frontier in science, SARS-CoV-2. So the question we try to address in what I'm going to present is what is the coding capacity of SARS-CoV-2? And I'll start by a short introduction about SARS-CoV-2. So it's a positive strand DNA RNA virus, 30 kb. And when the virus gets into the cell, the first orbs are translated, generating the RNA polymerase of the virus. And this polymerase will then synthesize the minor strand. But when doing it, it actually has a very unique feature that it can, when it gets to a specific sequence, which is called the TRS sequence, it can jump to the other TRS, which is located at 5' end of the genome. And this generates a series of subgenomic transcripts All of them share the same 5' UTR, but each one of them will encode for a different viral protein. And right now, the canonical annotations and the RefSeq annotation report on 12 viral proteins. So what we've done is use our expertise and perform ribosome profiling to look at exactly what's being translated in infected cells. So we infected Vero cells for 2 time points, 5 and 24 hours. And we also infected the Calu-free cells, which are lung human cells, for 7 hours. And each of these time points, we harvested the cells in 3 different ribosome profiling libraries. The first is treating the cells with cyclohexamide, which will freeze the ribosome wherever they are on the messenger RNA. And this generates a coverage along the gene. So here you see a meta-gene analysis of a sample. You can see the reads from the start until the end. And then we actually also have really nice phasing information, which shows how the ribosome moves every 3 base pairs in the mRNA that is translated. And then we also harvest cells using 2 drugs that affect only ribosomes that are now initiating translation. They work by different mechanisms, amitonin and leptidomycin. And then after we treat with the drugs, we give the cells few minutes, and the elongating ribosome will be chased out of the messenger RNA. And so we will be left with specific peak at the translation initiation site. And this is illustrated here. So you see with the arintoning and the leptidomycin, we get a very strong peak specifically at the translation initiation. And finally, we also did RNA-seq to get coverage on the RNA. And all of these experiments were done in biological replicates, which look really good. So here I'm showing you, single nucleotide resolution along the viral genome of the 2 replicates, just for an example of the LTM, that really looks amazingly well. Okay, so now we look at the virus, and what do we see? So first, I'm showing you all the canonical viral protein here labeled in different colors. And in green, you can see the RNA in log scale, and in red, the ribosome profiling the cyclohexamide sample. And you can see we see RNA increasing as we go to the 3' end, as one would expect, because of all of these just subgenomic RNAs that are accumulating. And if you look at the ribosome profiling data, you can see differences in the translation of different viral genes. So we can really quantify how much the virus is making from each viral protein. And this is illustrated here, both from the Vera cells and from the Calu-free infected cells. And you can see that the quantification looks largely similar. In both cells, the N protein, the nucleocapsid, is the most abundant proteins. But there are some variations. For example, in the Calu-free cells, we see much more in the M protein compared to the Vero cells. But what we can also do is look if there are anything else. Is there anything else that is being translated? And I wouldn't talk about it if we didn't find anything else. And so we actually were able to identify 23 novel viral proteins that are being translated. Many of them are upstream open reading frames. So I'm showing you here some examples. So here you can see ORF1A, which is the first ORF that is being translated. And you can see that there is a clear 2 upstream open reading frames that are translated upstream of it. And then there is another UORF, or maybe more interesting, another really interesting and mysterious translation initiation that we mapped in a CUG that is located at the position 60. And the reason it's interesting is that this CUG initiation, first, you can see that we don't see any downstream translation, suggesting it's maybe a ribosome that is pausing at this position and not really active translation. And this CUG is actually located in the leader sequence, which means that it's located in the sequence of all the viral RNAs, both the genomic and all the subgenomic RNAs. It's a little bit surprising. Why would you have a ribosome stuck upstream of all the transcript? So we looked more into this CUG, and we discovered that it's strongly enriched in reads that stem from the genomic RNA. So this initiation is not equally distributed between all transcripts. It mostly comes from the genome. And interestingly, a paper that did ribosome profiling at MHV, which is a mouse beta coronavirus, also identified the pause in a similar position. So what we think, and it's just hypothesis, is that maybe if on the genomic RNA, the ribosome is sitting at the CUG, maybe when we have a replication of the genome, the fact that the ribosome is sitting here, maybe it's masking the TRS of the leader and therefore helping the polymerase to achieve full replication of the full genome. Again, it's an hypothesis, but this, I didn't say this peak is really pronounced, and as you can see, the initiation of ORF1A is hardly seen when it's put in the scale of the CUG initiation. We also find versions of the canonical ORFs, so for example, ORF-M, the annotated initiation is here, and we found a 30 amino acid longer version of ORF-M. ORF10 is an ORF that there is a lot of discussion about whether it's a real viral protein. It's a small protein that's located in the most 3' end of the viral genome. We do see clear translation initiation in the beginning of ORF10, but it's really not efficiently translated. And actually the most dominant product of this locus is a 20 amino acid truncated version of ORF10 that is translated, you can see here.
Noam, it's 2 minutes.
Okay. Then actually the million-dollar question, did we discover any real or functional proteins? And we think we did, although we still need to work hard to prove it. So we found 4 out-of-frame ORFs that are longer than 30 amino acids, so potentially functional. One of them is actually ORF9b, which was annotated in SARS-CoV-1. So this is less surprising, but we can see it's really efficiently translated also in SARS-CoV-2. And 3 other internal ORFs are 2 internal ORFs that sits within ORF3A, and 1 that sits within the spike protein. The first one here and the internal spike are actually really highly expressed, as you can see in the densities in the black frame. And both of them are also predicted to be transmembrane proteins. And when we look at conservation, this ORF here, ORF3A, is actually highly conserved in all SARS-like viruses. There are now 3 different preprints that identify this ORF as an ORF that shows clear signature of purifying selection. So it's very likely this is a functional protein. It's 41 amino acids. It still needs to be studied whether this internal protein is also functional. I just want to emphasize that since we are talking about infection, even if a protein is not functional, it can still contribute to the immune response as it's likely presented in MHC+1. So the last thing we can look is actually compare the translation of viral protein to host protein. And what you can see is that, so this is the translation, this is the RNA level. We can see that the viral proteins are really taking over the cells, but this is mainly mediated by changes in the RNA. The RNA is really highly expressed, and that's why we see a strong translation. There is no changes in the translation efficiency of human genes compared to viral genes. So just to thank the people that did the work, this was led by 2 amazing, talented PhD students, Orel and Yara, with help from Aaron and Michal, and with tight collaboration with Nir Paran, the Israel Institute of Biological Research. Thank you. I'll be happy to take questions.
Thank you, Noam. Really great, exciting work. Got one question. With respect to these new ORFs, what do you think, now that we know about these, what are the ramifications for vaccine design and development?
So, I mean, one thing we are trying to see if these ORFs are generating any immune response, T-cells response. What's the meaning for vaccination? We don't know. I mean, the 2 that looks most promising with regard to potential function look as if they are transmembrane, so potentially they can be on the cell surface.
And then what about looking at other coronaviruses? I mean, can you make any inferences based on this and what is there in these other ORFs or these other viruses?
So we can do homology, right? So computational analysis, and this was already done. To actually do the experiments, if we have good collaboration, we can do this. But I mean, we need the virus and we need the facilities to do it.
Yeah, I could get you in touch with some people, I think. Here's a few more questions. Do you see any signature of ORFs in the minus strand?
That's an excellent question. We see protection in the minus strand, but from everything we look at, we don't think it's footprints, but honestly, we didn't look too deeply into this.
One more, is this CUG pause site found in all of the ORFs?
So the CUG is by definition sitting upstream of all the subgenomic RNAs. in most of the subgenomic RNAs, it will be an U-off. In one subgenomic RNA calling for the spike, if it would be initiating translation, it will generate an extension. But as I said, I mean, we see the footprints on all these subgenomic RNAs, but there is significant enrichment specifically to the genome.
Okay, so that's fantastic. So we're a few minutes behind, not that many minutes, but we're going to take a break. Let's do a 10-minute break and just reconvene at 9:25 with the last four speakers, and Saki will be chairing that. So I'll see you guys back here at in, I guess, now 9 minutes, or approximately 10 minutes. And thanks to all the speakers, really great session.
Thanks.
We are in the second session, and our first speaker is Danica Fujimori from QBI from San Francisco. Your first slide is already intriguing, so we are looking forward to hear you talk about the ribosome and antibiotics.
Thanks.
We'll move into the talk by Yaron Antebi from Weizmann for molecular genetics. And he will talk to us about promiscuity in information processing in cell-cell communication. Please, Yaron. Do we hear Yaron?
Yaron is on mute.
Hi. So it's great to be here, and I want to thank the organizer for this nice symposium. So I'll talk about the communication between cells, which is a key element in generating a coordinated response between multiple cell systems. And it does it because it can regulate many aspects of cellular behaviors. For example, it can regulate differentiation, proliferation, cell death, and migration of the cells. Interestingly, there are no specific signals that control specific aspects of the cell response, but rather cell-to-cell signaling is fundamentally combinatorial. There are many signaling pathways that combine together to control the cellular response, and each pathway actually utilizes multiple different ligands. What we're interested in my lab is to try and study how extracellular signals are being perceived by the cell, what intracellular picture the cells produce, and how this picture involved in controlling the cellular response. So today I'm going to talk on one of these pathways, the BMP pathway, which stands for bone morphogenetic protein. It's one of the most central developmental pathways, and it's actually found across all metazoans and responds to many of the developmental aspects. It was originally discovered by its ability to induce ectopic bone formation, but since then it was realized to control almost any tissue and many of the different biological processes. Interestingly, the pathway includes 15 different ligands and 7 different receptors that interact promiscuously with one another, and therefore it can combinatorially form almost 200 different complexes. But surprisingly, all of these complexes have the exact same biological effect. They all phosphorylate the same intracellular mediator. However, while this might seem to suggest that there's complete redundancy in the system, it is known that different ligands could have specific effects. Just as an example, in a specification of a sensory interneuron during early development, there are 5 different BMP ligands that are involved, and it was found that each of them has a specific effect of inducing different subtypes of interneurons. So this made us wonder, how can such promiscuous networks enable a ligand-specific response? And we start by generating a system where we could learn about how multiple ligands are integrated together into in this pathway, and what the role of the configuration of the receptors in controlling this integration. And then we use the system to enable a new way to differentiate between ligands based on their functional interactions. To do that, we use an epithelial cell line-based system where we integrated a transcriptional reporter controlling the expression of a yellow fluorescent protein in a BMP-dependent manner, and try to use it to describe all possible pairs of ligands of the BMP pathway. When we try to study the effect, we consider different concentration and combination of different concentrations of both ligands, and we found different ways of these ligands to interact together. In some cases, like what you see here, each ligand individually activate the cells, and also in combination in an additive way. But other ligands could show different responses. In some cases, ligands could actually inhibit a response by a first ligand, forming a situation where the overall response reflects the ratio between the two ligands. And finally, we also found an interesting situations where each ligand individually can activate a response, but when they are combined together, they inhibit each other such that overall the response seems to reflect some kind of an imbalance between the two ligands. In these ways, the cells can respond not just to an absolute amount of signals, but also to a composition and combinatorial information that's encoded in the composition of the signals. To gain further insight, we developed a mathematical model describing the competitive interactions between ligand and receptors. And we found two main results. First, we were able to generate 4 distinct response types based only on these competitive interactions. 3 of these responses simply reflects the responses I showed you experimentally, but the model also suggests the possible 4th behavior where the response is maximal only when both ligands appeared in some balanced way. In addition, we can start to ask what happens when we consider cells with different receptors. And in this case, we can see that the system is very versatile and can produce distinct responses and actually interpolate between all 4 distinct behaviors for different cell types expressing different receptors. This was also verified experimentally. As we go back to our experimental system, we can change the receptors either exogenously or by considering different cell types. And we can see that the same ligands can be integrated either additively, ratiometrically, or in a balanced way. So now we can try to quantify this behavior across different ligands. And for reference, we can consider the response from a single ligand or from the other one, and the sum of the 2 responses. We can then compare these reference points to the actual measurement of the response to the combined signal, and we can generate 4 different regimes. These combined signals could be much weaker, like the imbalanced behavior. It could be intermediate, like what we saw in the ratiometric case. It could be additive, or it could be much stronger, like in the balance effect. We therefore used behavior coefficients that quantify this range from -2 to 2 and give us some indication of the overall response and overall interaction between a pair of ligands. We now need to measure this behavior coefficient across all possible 45 pairs of the different ligands. And I remind you that each square is actually composed of multiple measurements.
2 minutes. 2 minutes.
Across many different concentrations. To do that, we used an automatic robotic system that allows us to measure hundreds of different concentrations at a time. And we can now extract the different behaviors for all these pairs, where each row here represents kind of an interaction fingerprint between every ligand and all the other ligands in the system. This allows us to cluster the similar ligands together and actually generate an equivalence map or a redundancy map that's showing that in this epithelial cell line, it can distinguish between 4 different classes of BMP ligands. As an interesting note, I would say that we saw that receptors could affect the way cells interpret signals. And indeed, if we consider the same map for another cell type, embryonic stem cells, we get that they can actually distinguish between more types of BMP signals and have a higher resolution in identifying specific BMP identity. One final comment, we can use this to ask what is a specific effect of receptors on the ability of cells to distinguish these ligands. And here I consider the BMP-R1B receptor. It's a relatively rarely used receptor. And when we overexpress it in our cell lines, we find that almost all difference between ligands disappear and most ligands are completely redundant. Interestingly, this is used in joint formation, which have 2 main regions, the interzone, which needs to respond in a very specific way and different for different BMP ligands, and the external regions that generally responds for all BMPs. And indeed, we can see that the more general responders express these receptors that actually allows them to respond universally to all BMP ligands. So with that, I want to summarize. We find that BMP and the promiscuous nature of the system allows more complex computations and allow the cells to respond to specific combinations of signals. This combinatorial response allows us to define functionally different groups of ligands and start to decipher how specific receptors can control the way cells are able to distinguish and specifically respond to different signals. I want to thank the people in my lab here at Weizmann, and some of this work was done in collaboration with the Elowitz lab at Caltech. And thank you.
Thank you, Yaron. That was really interesting. Let's wait for some questions to appear. There is one. What is known about the molecular mechanism underlying the different archetype responses?
So from the model, the entire mechanism could arise only from the interaction, the competitive interaction of receptors and ligands. And we can actually get some insight about the different behaviors. For example, ratiometric could be is generated through a simple competitive inhibitory effect. And the more complex behaviors like the imbalance are actually a more complex variant of these effects, where the main, interestingly, the main effect arises from the notion that the affinity, the binding affinity between ligand and receptors does not need to correlate with the phosphorylation efficiency of the receptors. And the interplay between these 2 types of parameters produces the more general responses.
I don't see an additional question, so I'll follow up on this. Is it possible that the ligands also operate on one another directly? Maybe one degrades the other or potentiates the other?
So we didn't rule it out, but from what's known about the system, there are some extracellular components that could interact with the ligands, but it's not known that the ligands themselves inhibit each other. And additionally, most of these effects can be reprogrammed or controlled by only changes to receptors on the surface of the cell. So it definitely cannot completely explain all of these effects.
We have 30 seconds, so if there's no more, are there ligands that are across many other ligands participating in a particular response, like ratiometric or others, or every pair does something different? Maybe very briefly, if you can answer.
So generally, most of the interactions are ligand dependent. So even if there are some inhibitory effects, it's ligand dependent inhibitory effects. There are ligands, BMP10 and BMP9 are two ligands that are specifically associated with more positive for BMP10 and for BMP9 and more negative for BMP10 interactions. So they might have a more universal positive and negative roles.
There's another question that just came, but I think we are running out of time, from Zev Gartner, so maybe you can respond to that later. And without further ado, we'll move into the talk. So thank you, Yaron. And we'll move into the talk of Tanja Kortemme, and she will talk about cellular consequences of allostery. Please, Tanja.
Okay, let's see. Can you see my screen?
Yeah.
All right, so my lab normally works on computational protein design, but today I'm going to share a very different story where we discover several new allosteric sites in a central biological regulator, and making specific perturbations at those sites has widespread cellular consequences. And this story was really spearheaded by Tina, collaboration with Nevan and Mark at UCSF, and in particular, Jiwei, Gwen, and Danielle in Nevan's lab. So the central question here that we were asking is, how do proteins control functions in interconnected and often rather complex cellular networks? And so while we know for many proteins, really in a lot of detail, how they function biochemically, it is often much more difficult to translate this detailed biophysical knowledge of protein function to how a protein really operates in the much more complex context of all its interaction partners in a cellular system. And so the specific model system that we picked to interrogate this broad question is a simple biological switch that has two states, an on and an off state, so a two-state switch, where the two states are switched with a pair of regulators. And of course, this biochemical switch is then embedded into the context of its cellular interactions in the cellular context. Today, I'm going to talk about two different questions. The first one is the question of functional multispecificity. In our case, our biological switch regulates multiple biological processes distinctly. So how does a single switch achieve that multispecificity? The way we're going to be studying our biological switch is by perturbations, in our case, specific mutations. And the question is, if we perturb the system with these specific perturbations, are the effects isolated to the site of mutation, or are they propagated much more widespread within the context of the entire cellular network? In terms of approaches, what we're going to be doing is we're going to be making more than 50 single amino acid point mutations to the biological switch. And then we're going to be studying the consequences of these perturbations on the cellular level by looking at quantitative genetic interactions in our model organism Saccharomyces cerevisiae. We're also going to look at the changes of these point mutations to the physical protein-protein interaction network in the cell using affinity purification mass spectrometry. And we're also going, on the side of molecular biophysics, going to look at the effect of the mutations on the kinetics of interconversion between the two states of the switch, as well as on conformational equilibria that we study by nuclear magnetic resonance spectroscopy. And then in the end, I will try to come up with a model that links the molecular and the cellular effects. Our specific example of the biological switch is a GTPase that, like all GTPases, has a GDP and a GTP bound state and is regulated by the two central regulators, a GEF and a GAP. And our specific GTPase is called RAN in mammalian systems, or GSP1 in yeast. We design our perturbations to the switch by making point mutations in the GTPase in interfaces with specific interaction partners of the GTPase based on structural information. We don't make any mutations in the actual active site of the GTPase or anywhere close to the switch regions. We make all our point mutations on the yeast chromosome, and as far as we can tell, these mutations do not significantly affect protein stability, both with purified proteins as well as in the cellular context. And then, of course, the simple prediction was if we perturb the interactions with specific biological partners of the GTPase, this might lead to specifically affected differential biological functions. The way we characterize the functional effects of the GTPase is through quantitative genetic interaction mapping or in collaboration with Nevan's group. And basically what we get for each of our point mutations is a vector with 1,444 numbers quantifying the genetic interactions of the mutant with one of each of more than 1,400 genes deleted individually in yeast. The way to generally analyze these types of genetic interactions is to look at these vectors as functional profiles that we can compare to each other using correlation analysis. So we also have similar types of data for deletion of around 4,000 yeast genes. And so what we can do is we can correlate these functional profiles between the different mutants or between point mutants and genes, and high profile correlations indicate shared affected biological functions by each of these perturbations. So this is what the data looks like. About half of our designed GTPase point mutations have significant genetic interactions across this panel of genes, as shown here by these EMAP scores, significant in yellow and blue. And then when we cluster these genetic interaction profiles, these functional profiles, the functional clustering makes sense. For example, the genes involved in spindle assembly regulation cluster together, and that is one of the biological functions of our GTPase. So now what we wanted to see is whether if we now correlate a point mutation in the GTPase in an interface with a partner with deletion of that partner, then we should see a significant functional correlation saying that the point mutation affected the interaction with that partner and therefore the biological function. And so we quantify profile correlations between mutants and genes now in two different cases. I'm going to show you two different distributions. One where we correlate a point mutation with a deleted gene where the point mutation is not in the interface with that protein, and the other distribution is where the point mutation is in the interface with that protein. And of course, the prediction was that we see significant correlations on the right, but not significant correlations on the left. However, what we found is that the two distributions of correlation coefficients are identical. So that was a surprise here, that it seems that the interface position of mutation in the GTPase is not a good predictor of the functional outcome of that mutation based on the genetic interaction profiles. So then, of course, we needed to understand, do the mutations indeed affect the targeted physical interactions? And there's an obvious experiment that we can do where we can look directly at the effect of each mutations on the physical interactions that the mutated protein makes relative to the wild type protein using affinity purification mass spectrometry. So again, I'm going to show you two distributions. One is where the mutation is in the partner interface and one where it's not, but now looking at the mass spec data. So as expected, when the mutation is in the interface with the partner, overall, this is the zero line here, overall, the abundance of that pull-down partner in the APMS experiment is reduced. So that is just like we expected, we affect the targeted interactions. However, now when the mutation is not in the interface with the partner, we expected this distribution to be around 0, to not see any significant effects. However, we see the opposite. We see that there are large effects, both in terms of decreased interactions as well as in terms of increased interactions. So it seems that what the point mutations are doing is they're leading to widespread rewiring of the physical interaction. So in trying to think about what could be a possible alternative explanation for this widespread rewiring of the physical interaction network, we turned to molecular biophysics and we asked the question, well, could these changes in the genetic and in the physical interaction profiles be explained by effects of the mutation that are directly on the cycling of switching the biological switch between its on and its off state? And we're going to look at this using both enzyme kinetics of the switch as well as I said, conformational equilibrium. So I'm going to show you the effects of the point.
2 minutes, please.
I'm going to show you the effect of the point mutation on the kinetics first. And so up here, we're going to be looking at effect of point mutations on the nucleotide exchange kinetics and down here on the GTP hydrolysis kinetics. This is a lot of data, but illustrating that essentially all of our mutations, if you look at the wild type levels up here and down here in the dotted lines, essentially all of our mutations affect either the GEF-mediated nucleotide exchange reaction or the GAP-mediated GTP hydrolysis. And what was particularly surprising to us, particularly in the case of the GAP-mediated GTP hydrolysis, that only one of all of these mutations that I'm showing here was actually directly in the interface, oh, this is a typo, in the interface with the GAP, suggesting that the mutations indeed, as I mentioned at the beginning, act by an allosteric mechanism. So to confirm whether we actually see any structural evidence for the allosteric changes here, we turn to a rather simple experiment, one-dimensional P31 NMR. And I'm going to show down here the spectrum, a one-dimensional spectrum of the wild-type protein, where these two peaks here are indicative of two different conformational states of the GTPase in the active site. And you can see here for the wild-type protein that most of the protein is in this state 2. However, we repeated the same experiment now for a range of different mutations that I showed you before, and you can appreciate that for the mutations down here, almost everything shifts into the state 2 conformation, and if you look at the mutations up here, almost everything shifts to the gamma-1 conformation. And what I'm indicating in red here are all positions that are at least 18 angstroms away from the active site, suggesting that these mutations act allosterically, directly affecting the conformational balance of states in the active site of the GTPase. So then, of course, bringing everything together, the question is, can this altered balance of the GTPase switch cycle explain the genetic interaction profiles? So what I showed you before is the genetic interaction profiles grouped by function, which made sense, but they did not group by interface location of the mutation, which was puzzling. And so the question now is, now that we have the kinetic effects, do the profiles group by the effect of the mutation on the kinetics of the switch cycle? And I'm almost done. So when we do that, now the first grouping that actually gives us a difference is we're grouping the mutants into the ones that mainly affect the hydrolysis capacity, or the mutants that mainly affect the nucleotide exchange. And we find 3 groups of genes. For group 1, they are correlated significantly with both sets of mutants. But now when we look at 2 genes, they are primarily correlated with mutants with reduced hydrolysis. And when we look at group 3 genes, they are primarily correlated with mutants with reduced exchange. And this actually has biological consequences, and this is my last data slide, that we can now group different biological processes regulated by the switch based on the kinetic sensitivity of these parameters, where genes in nuclear transport are sensitive to mutations affecting both sides of the cycle, so they're sensitive to the balance. Genes, for example, in spindle assembly regulation, are primarily sensitive to mutations that affect GTP hydrolysis, so they're sensitive to the ability of the switch to turn off. And genes involved in tRNA modification are primarily sensitive to the ability of the switch to turn on. So to bring it all together, the point mutations that we see in our central biological switch have widespread biological effects, not just localized to individual interactions in the network. We find that this is mediated by allosteric effects affecting the GTPase switch, and that the multispecificity of the single switch could be explained by the differential sensitivity of different biological processes to the precise control of the switch cycle kinetics. And going forward, I am very interested in exploring algorithms that in a similar fashion could integrate detailed biophysical analyses with systems level processes for other biological systems. Thank you.
Thank you, Tanja. Thank you very much. Open for questions.
So I see a question from Nevan here. What is the next system you want to apply this to in humans? So I think on a fundamental level, it is extremely interesting to actually use the same GTPase, but now with its potentially much more complex regulation that is imposed in the human cell system relative to the yeast system. I think that other two-state switches, to my mind, if you think about the biological mechanisms here that are very attractive to study, are other types of two-state regulators like kinase phosphatase systems, methylase, demethylase systems, and so forth, that I think all have the same potential of being highly sensitive to the opposing action of these regulators.
Good. And Giliona says, I may have missed this, but what type of perturbations have you used?
Yes, these are, sorry, this is fast in 10 minutes. So these were single amino acid point mutations at the surface of the GTPase, but away from the active site. And they were integrated into the yeast chromosome to preserve the regulation as much as possible. And it would be very interesting, actually, if I can talk about more things that I would like to do with this. And now it would be very interesting to make quantitative perturbations to the activity or the level of the regulators to directly test this model, to what extent the regulators tune the kinetics of switching.
So as a quick follow-up, he asks, no perturbation in the growth, if you can be very brief.
Yeah, what's very brief here, the perturbation of growth is the readout of the quantitative genetic interactions.
Right. Okay, thank you so much, Tanja. That was really interesting. And the last speaker is actually myself. So I'm Tawfik Pilpel from Weizmann. And I hope you see my screen. So I can't thank the organizers, but I definitely thank all of you for coming, and Nevan and the QBI guys for initiating this. And what I want to talk to you today is about a project that started recently in the lab that asks questions about variation in mutation rate. In fact, it's going to be a merger of two projects, time permitting. So I think we all appreciate that evolution proceeds through mutations. No mutations, no evolution. On the other hand, if you have too many mutations, of course, Many of them are deleterious. And a very basic question in molecular evolution is, what is the optimal mutation rate for an organism? And this is just one example of people measuring and finding an interesting correlation between mutation rate and genome size. Different organisms have different mutation rates. But what's common to all those measurements and assumptions, etc., is that there is one mutation rate for an organism, but in fact, there could be very interesting deviations from that. One known deviation from a constant mutation rate is, of course, stress-induced mutagenesis. This is a well-known phenomenon. Under stress, organisms increase mutation rate, and they appear to do that in a regulated and an adaptive fashion. But variability in mutation rate can manifest itself not only when times shift from being comfortable to less comfortable, but for instance, what if different individuals in the population operated under different mutation rate? Is it possible that some individuals in the same population that are not different genetically from the others would have a higher mutation rate than others? And another axis at which a mutation rate can vary is for the same individual across different genes in the genome. So is it possible that not all genes evolve under the same mutation rate, but rather some are programmed perhaps to evolve more rapidly than others? So in order to ask those questions, we decided to look into a very well-known and a slightly less well-known aspect or branch of systems biology. The first has to do with noise in gene expression. So I think every systems biologist appreciates that there is noise in gene expression. This goes back to the classical papers by Elowitz et al. This is different E. coli cells genetically identical, expressing though GFP to different levels, suggesting that stochasticity plays a role in gene expression. We and Jonathan Weizmann from UCSF have shown across many genes, natural genes of the genome, that they present variation. This is if you plot yeast genes, their mean abundance against the noise, you see an interesting scaling, but here above the major scaling, we see the stress-induced related genes, or stress genes in particular, under stressful conditions, they show a lot of noise. So stochasticity appears to be regulated within cells. Can we use that to say something interesting about mutation rate? Well, before we answer that question, another source of stochasticity, if the previous was amount stochasticity, the same protein expressed in different levels, what we have recently discovered is that there is another major source of stochasticity, and that's sequence stochasticity. What I mean by sequence stochasticity is that the molecular machinery that produces RNAs and proteins within cells is not perfect. Ribosome and many other components do mistakes. So in this paper, we recently teamed up with several colleagues to quantify the extent of mutations and mistakes that are seeded in proteins while translated. And in similarity to the previous observation made here at the quantitative level, we found that different proteins in cells are produced with different levels of mistakes. I don't have time to go into the details of how we did that, but it's all based on quantification of intensities of peptides in mass spectrometry. We identify perfectly translated peptide and peptide translated with mistakes. And if you plot the expression level of proteins against the mutation rate, you see an interesting decline. But what was more interesting is that if you look at some of the genes that are definitely above the trend, you see, for example, DNA repair enzymes. These are proteins that are reducing mutation rate in DNA replication. And what we see here that they appear to be deliberately prone to be translated with mistakes. So what could be the consequence of that? Well, it appears that we have post-dicted a paper that was published about 30 years earlier by Nino that actually predicted that most mutators within cells are going to be, as he called it, transient mutators. So we used to think about mutators as strains that have a mutation that renders them more highly mutagenic. Nino predicted, and we have recently shown in this experiment, that actually most of the mutations, most of the mutator effects that reside within cells are actually due to mistakes in translation and in transcription of DNA repair enzymes, rendering some of them less or more active in different times. So this all sets the stage from the quantitative point of view and from the sequence mistake point of view to the main question that we wanted to ask, and that is, what is the variation in the population in mutation rate between individuals? So another source of inspiration was this paper from Johan Paulson et al., in which he looked at the ADA protein, a DNA repair enzyme in E. coli. And when he measured single-cellularly the expression level of ADA in individual cells, he noticed that some cells have ADA and others don't. And not only that, ADA operates on a positive feedback loop, so that if one cell had many copies of ADA, the offsprings of that cell would tend to have more than a cell that has none or very few. So that led Gabriela, a PhD student in my lab, to ask, what is the optimal mode of mutation rate inheritance within cells? Is the ADA protein presenting to us an optimal strategy in which different cells split the job between them? Some that have a high level of the protein would operate on low mutation rate, and others that have a low level of the protein, this is a DNA repair enzyme, if you have low from it, you'll have many mutations. So we are asking two questions. First, what's the meaning of having variation in the mutation rate between cells in the population, due, for example, to having different levels or different amount of mistakes in the generation of that protein? And a second question, what is the optimal way to inherit this variation to the next generation? So we are doing experiments at the moment, very computational and mathematical. In future, we hope to do experiments also in reality, in which we have mathematical organisms that are evolving on fitness landscapes, and their job is to get to the peak of the landscape. And they operate with variation in mutation rate, so that within a population, although they are genetically identical, the mutation rate varies. Some are inherent, some are more noisy, some are more accurate than others. But the most important thing that Gabriela experiments with is different regimes of inheritance of the mutation rate. So if you are a mutator, that means that your tendency to make mistake is genetically encoded within you. You will have a high or low mutation rate, and the offsprings would inherit that mutation rate precisely. If noise is totally randomly inherited between generations, if you had a high mutation rate, your offsprings could have a very low mutation rate and vice versa. And if something like the ADA regime is at work, namely there is some epigenetic inheritance of the mutation rate, namely if you had a high mutation rates, your offsprings are likely to have a higher mutation rate, but not precisely as you are, then you are in some kind of a noisily inherited mutation rate regime. That's the red here. To make a long story short, we let our mathematical organisms solve these fitness landscapes, and we ask, how does their fitness increase with time? So of course they are all improving, but the main important result here is that those that inherit, those that have variation in mutation rate, and that they inherit that mutation rate variation in a semi-stochastic way, in an epigenetically inherited mechanism, appear to perform the best. So what happens there, when we look at the mutation rate throughout time, in the noisily inherited mutation regime and in the perfectly inherited mutation regime, we see that when mutation rate is inherited perfectly, there is a phase of increasing mutation rate. This is needed in order to climb fitness landscapes, but once they get to some optimum, they are stuck at that mutation rate. In contrast, the population that have the noisily inherited mutation rate features a very interesting, although we didn't tell them to do that, features a very interesting abrupt phase of increasing mutation rate. This allows them to climb into fitness landscapes, but they are not committed to stay with those high mutation rates because it's noisily inherited. And once they get to the top of the mountains, they are replaced by their progeny that are featuring, just by stochasticity, a lower mutation rate. So I want to conclude, I didn't get to the second part. I want to conclude by thanking people, but before that, let me just wrap it up and say that what we are beginning to discover is a very interesting regime of division of labors. Some in the population operate on a very high mutation rate. Some operate on a very low mutation rate, and this tendency is inherited, but semi-accurately or epigenetically, if you will, not genetically and not stochastically. And that appears to be, at least mathematically and with simulations, and hopefully soon in experiments, that appears to be an optimal strategy for a population to solve complicated fitness landscapes. So I want to conclude by thanking Gabriela and Yoav Ram, our collaborator. I didn't get to show the work of Sivan and Yonat, and Orna is, of course, very much involved in all of this too. So thank you. And Nevan, are you going to moderate a discussion for me?
I am here. Okay, so Tanya's got a question. She's excited about your talk. How will you test this experimentally, Saki?
Yeah, that's a good question. We are contemplating two ways to test that experimentally. We need to generate either yeast cells or bacterial cells in which we will manipulate one or two of the following properties, or actually three properties. One is that we want to generate, you remember that ADA protein that I described to you, this expressed in a very noisy fashion across cells. We want to evolve E. coli cells to feature low noise level in ADA. Ideally, everyone would have the same amount of ADA. So now you don't have the variation in mutation rate to begin with. There's no partition into explorers and exploiters, if you will. So that's one potential manipulation. And we have, I think we may have a strategy copied from Shooting You, a postdoc in the lab, in order to do that. An alternative is to go directly according to the, following the prediction of the mathematical model and manipulate not the noise level itself, but the mode of inheritance of the noise to the next generation. So with the ADA protein, the inheritance appears to be kind of epigenetic, not very accurate and not very sloppy. We are thinking about various ways, but this is very, very tricky, various ways to manipulate the cells in order to inherit either very accurately or very not accurately the expression level. And lastly, I've shown to you that, that from the project on translation mistakes, I've shown to you that DNA repair enzymes are translated with mistakes. That's a very interesting quality that we discovered that they have, and as I said, it was predicted 30 years before that. It turns out that codon usage, for example, could determine the amount of mistakes that the ribosome would do when translating a protein. So perhaps another possibility is to go into those DNA repair enzymes, convert their codons synonymously, and get either a more accurately translated or less accurately translated DNA repair enzyme, and again, let the organism evolve towards various challenges and see what it does.
Okay, we got, yeah, that's great. Felix Jonas also enjoyed your talk. The question is the timescale of the epigenetic mechanism dependent on the shape of the fitness landscape?
If it depends on the fit, I guess, yeah, it's a good question. I guess that for different fitness landscapes, there would be a different optimum of the timescale of the epigenetic inheritance. The fastest epigenetic inheritance mechanisms are very simple. You have a protein, it's present in a cell in many copies, and then when the cell splits, when the cell divides, each cell gets roughly half of the amount, but not exactly. So that's a very sloppy mechanism of inheriting epigenetically information, and its mixing time is the fastest. Others that are based on chromatin modifications, etc., are probably more long enduring. I tend to think, and I think it's a very interesting question, that the more rugged fitness landscape versus the smoother fitness landscape might enjoy differently, either from long-lasting epigenetic propagation versus short-lasting ones. But actually, I think if Gabriela is here, I think it would be interesting to test that at least first in the models and perhaps later with the experiments.
Okay, so we're 1 minute to 10:30. Thanks, Saki. I'd like to thank everybody for the fantastic set of talks. I'm very much looking forward to next week's episode. I don't know if Saki or Danny had any wise words here, or Tanya, to wrap up before we close down?
Yeah.
Oh, there's the bell. Okay, so thanks everybody for today, and we will see you next week, same time, for the next set of 8 talks.
Thank you, Nevan, Jacqueline, and everyone.
Thanks. Nice
Thanks,
to
guys.
see you
Thank you.
guys. Bye.
Thank you. This is terrific.